Okay, join to this link. Am I audible? Shall I start? So last time what we have seen, the secondary active transport. What is secondary active transport? We have seen the example of secondary active transport, that is the lactose permease. What is secondary active transport? Transport of molecule across the cell membrane utilizing energy other than the ATP. Is it? Do you remember? Instead of giving your presentee here, don't give your presentee. Instead of giving your presentee, you pay your attention here. In the last lecture, what we studied? Secondary active transport. What is secondary active transport? Transport of molecules across the cell membrane utilizing the energy other than ATP. Means primary active transport utilizes ATP as the energy source. For the movement of molecule with the help of primary active transport, ATP acts as an energy source because it is against the concentration gradient. Against the concentration gradient means from lower concentration to higher concentration. So it requires ATP as the energy source. Now secondary active transport is also against the concentration gradient, means from lower concentration to higher concentration. But energy which comes in the secondary active transport is the energy from the electrochemical gradient created ड्युरिंग द प्रायमरी ऍक्टिव्ह ट्रान्सपोर्ट. म्हणजे प्रायमरी ऍक्टिव्ह ट्रान्सपोर्टमध्ये जी काही एनर्जी किंवा जो काही इलेक्ट्रोकेमिकल ग्रेडियंट निर्माण होतो, the electrochemical gradient which is generated in the primary active transport, it is used as the energy source for the secondary active transport. म्हणजे प्रायमरी ऍक्टिव्ह ट्रान्सपोर्ट हे एटीपीच्या मदतीने होईल, पण प्रायमरी ऍक्टिव्ह ट्रान्सपोर्ट होत असताना जी काही एनर्जी क्रिएट होणारे दॅट एनर्जी विच इज जनरेटेड or directly energy is not generated, electrochemical gradient is generated. म्हणजे एकीकडे कॉन्सन्ट्रेशन हायर आणि एकीकडे लोअर, असं जे इलेक्ट्रोकेमिकल ग्रेडियंट क्रिएट होणारी दॅट इज यूज फॉर द एनर्जी जनरेशन, दॅट इज यूज एज द एनर्जी सोर्स इन द सेकंडरी ऍक्टिव्ह ट्रान्सपोर्ट. आर यू गेटिंग मी? समजतंय का? आपण सेकंडरी ऍक्टिव्ह ट्रान्सपोर्टचे इम्पॉर्टंट कॅरेक्टर्स मागच्या लेक्चरला बघितले होते, परत आज पण मी रिवाइज केले. What is secondary active transport? It is the transport against the concentration gradient with the help of energy other than the ATP energy. So whatever the prime energy generated because of the electrochemical gradient of primary active transport is used as the energy source for the secondary active transport. Okay. So this transport takes place in the same direction or in the opposite direction. आता सेकंडरी ऍक्टिव्ह ट्रान्सपोर्टचं काय वेगळं कॅरेक्टर आहे प्रायमरी ऍक्टिव्ह ट्रान्सपोर्ट पेक्षा? सेकंडरी ऍक्टिव्ह ट्रान्सपोर्ट मे टेक्स प्लेस इन द सेम डायरेक्शन, दॅट इज सायमल्टेनियसली ट्रान्सफर इनसाइड द सेल, और इट मे टेक्स प्लेस इन द अपोजिट डायरेक्शन. अपोजिट डायरेक्शन म्हणजे काय? एखादा कंपोनंट सेलमध्ये जाईल तर सेलमधनं एखादा कंपोनंट बाहेर पास होईल, that is in the opposite direction also or in the same direction. So it is secondary active transport port is also known as ion coupled transport. Ion coupled transport म्हणजे coupled transport आहे. Coupled means either movement of molecule in the same direction or in the opposite direction, but the two molecule move simultaneously in the secondary active transport, hence it is also known as ion coupled transport. So last time we have seen the example of secondary active transport, that is the lactose permease. Lactose permease is the group of transport protein. हे बघा, हे तुम्हाला दाखवलं होतं मी lactose. This is the lactose permease, which is also known as the galactoside permease, which allows the passage of lactose from outside to the inside of the cell. Lactose permease is a protein molecule which is present in the सेल मेम्ब्रेन. हे जे आहे स्ट्रक्चर हे सेल मेम्ब्रेनचं आहे. आर यू एबल टू सी दिस स्लाइड? And this lactose permease is able to transport lactose molecule from outside of the cell to the inside. But during this passage of lactose, these H plus ions, they are also passes inside the cell. सेल मध्ये त्याच्याबरोबर कोण जातंय? H plus पण जात आहेत. एच प्लसचं कॉन्सन्ट्रेशन सेलमध्ये वाढलं तर काय होईल? एच प्लस अगेन पासेस आउटसाइड द सेल बाय दिस प्रोटॉन पंप. या प्रोटॉन पंपनी हे एच प्लस आयन पुन्हा आतमधून बाहेर पास होतील. पण लॅक्टोज परमिएस, व्हॉट इज द रोल ऑफ लॅक्टोज परमिएस? लॅक्टोज परमिएस इज अ सिम्पोर्ट मेकॅनिझम. सिम्पोर्ट टाइप ऑफ ट्रान्सपोर्ट. सिम्पोर्ट ट्रान्सपोर्ट म्हणजे काय की इट ट्रान्सपोर्ट लॅक्टोज अलॉंग विथ द एच प्लस आयन्स, म्हणजे एच प्लस आयन्स पण त्याच्याबरोबर ट्रान्सफर होतात विथ द हेल्प ऑफ दिस प्रोटीन known as galactoside permease or lactose permease. Are you hearing me properly? Instead of giving your attendance, present madam, are you hearing me properly? That tell me. Your attendance, I will conduct the assignment, and after that assignment I will write down your presenting. Okay. So lactose permease is the important example of secondary active transport. Lactose permease is the proton driven co-transporter which is present in E. coli. E. coli मध्ये लॅक्टोज परमेस पहिल्यांदा शोधून काढला. So it is present in E. coli as a proton driven co-transporter, म्हणजे प्रोटॉन पण त्याच्याबरोबर H+ आयन पण सेलमध्ये पास होतात. It functions as a monomer to transport one proton and one lactose molecule inside the cell. This function, lactose permease function as a monomer to transport one proton molecule and one lactose molecule inside the cell with the net accumulation of lactose. Net accumulation of lactose means only lactose is accumulated inside the cell, while the proton which are accumulated inside the cell, they again with the help of proton pump, with the help of this proton pump, that proton molecule, that is H+ molecule, again come outside the cell. ते पुन्हा सेलच्या बाहेर निघून येतात, कारण increasing concentration of H+ ions is again toxic to cell. So this H+ ion again come outside. So only there is a net accumulation of lactose inside the cell with the help of this lactose permease system. Now next is the sodium glucose symport. This is our today's new topic, that is the sodium glucose symport. Sodium glucose symport is again co-transport system. It is co-transport system. It utilizes sodium gradient to drive the uphill transport of glucose. म्हणजे इथे इम्पॉर्टंट आपल्याला कोणाला ट्रान्सफर करायचं आहे, ग्लुकोजला. लॅक्टोज परमेस कोणाला ट्रान्सफर करतं, लॅक्टोजला. सेल utilizes lactose or glucose as the source of कार्बन सोर्स, एनर्जी सोर्स म्हणून सेल ग्लुकोज किंवा लॅक्टोज वापरतात. मग हे ग्लुकोज किंवा लॅक्टोज सेलमध्ये पास करण्यासाठी सेलला त्याच्याबरोबर दुसरे आयन्स पण मध्ये पास करावे लागतात. सो इन सोडियम ग्लुकोज सिम्पोर्ट, this is again a co-transport system where sodium ions means sodium gradient passes the transport of glucose inside the cell. Sodium gradient to drive the uphill transport of glucose त्याच्याबरोबर मध्ये पास होतात. It transport one molecule of glucose into cell for every 2 molecules of sodium ion. म्हणजे 2 सोडियम आयन मध्ये जातील तर त्याच्याबरोबर एक ग्लुकोजचा मॉलिक्यूल पण राहील. हे बघा, हे डायग्राम बघा. दिस इज सोडियम ग्लुकोज सिम्पोर्ट. ग्लुकोज मॉलिक्यूल पण सेलमध्ये जातोय, सोडियम मॉलिक्यूल पण जात आहेत. 2 सोडियम मॉलिक्यूल पासेस इनसाइड द सेल. 2 सोडियम मॉलिक्यूल सेलमध्ये जातात. Okay, so it transport 1 molecule of glucose and 2 molecule of sodium ions in the cell. So this symporter is located into the small intestine, in heart and in brain. It is located in the proximal tubules, in each nephron, in the kidney. आणि कुठे असतात, किडनी मध्ये असतात. This sodium glucose symport mechanism, these pumps are located in the small intestine, in heart, in brain and in the proximal tubules in each nephron in the kidney. Kidney is made up of millions of nephrons. Kidney मध्ये काय असतात, millions of nephrons असतात, and this nephron consist of the proximal tubules where this sodium glucose symport is located. मग ते काय करतात, they contribute in the renal glucose reabsorption. म्हणजे सपोज युरिन मधनं जर ग्लुकोज पास होत असेल तर त्या ग्लुकोजचं रिअब्सॉर्प्शन करतात कोण, दिस सोडियम ग्लुकोज सिम्पोर्टर्स or protein present in the proximal tubules of the nephrons of the kidney. किडनी मध्ये जे काही आहे हे सोडियम ग्लुकोज सिम्पोर्ट मेकॅनिझम ते ग्लुकोजला रिअब्सॉर्ब करतो. ओके, फक्त इन केस ऑफ डायबिटिक पर्सन एक्सेस ग्लुकोज इज thrown out of the body because of the person is diabetic, that is the person have the excess glucose in the blood, and that blood glucose is passes, means it is thrown out of the body through the urine मधून ते त्याचं डिस्कार्ड होतं. पण जर हेल्दी इंडिव्हिज्युअल असेल तर त्याच्यामध्ये ग्लुकोजचं पुन्हा रिअब्सॉर्प्शन होतं. सो दिस इज सोडियम ग्लुकोज ट्रान्सपोर्ट, दॅट इज द सिम्पोर्टर मेकॅनिझम. काय होते बघा, ग्लुकोजचं सेलमध्ये येतं, ग्लुकोज सेलमध्ये आल्यानंतर ग्लुकोज एनर्जी सोर्स म्हणून वापरला जातो, आणि सोडियम आयन जे आहे ते सोडियम आयन परत सेलच्या बाहेर पास होतात विथ द हेल्प ऑफ सोडियम पोटॅशियम ATPase पंप. नाहीतर मग काय होईल, सेलमध्ये सोडियमचं कॉन्सन्ट्रेशन वाढेल, मग सेलमध्ये सोडियमचं कॉन्सन्ट्रेशन मेंटेन राहावं म्हणून आपण सोडियम पोटॅशियम ATPase किंवा सोडियम पोटॅशियम पंप याचं फंक्शन आपण अगोदरच पाहिलंय. We have learned about the sodium potassium pump or sodium potassium ATPase. See, we have seen the mechanism of sodium potassium ATPase or sodium potassium pump, which is the active transport system. It is responsible for maintaining high potassium and low sodium concentration inside the cell. If the concentration of sodium ion in the cell increases, it leads to the cell lysis. मग हे सोडियमचं कॉन्सन्ट्रेशन सेलमध्ये वाढत राहतं अशा वेगवेगळ्या प्रोटीन्समुळे. मग ते मेंटेन कोण करतं, तर दिस सोडियम पोटॅशियम पंप. आपण हे आताच मागच्या लेक्चरमध्ये शिकलो होतो. ओके. सो again the increased concentration of sodium ion, these are passes out of the cell by the sodium potassium ATPase. When the sodium ion passes outside, there is a simultaneously intake of potassium ion inside the cell or simultaneous passage of potassium ion inside the cell. Okay, and this sodium potassium symporter, sodium glucose symporter, the sodium glucose symporter or transporter, it transport glucose molecule inside the cell with the sodium molecule simultaneously inside the cell, which is again passes outside the cell. Okay, so next is the carrier proteins. So carrier proteins are the transporter proteins or transporters that binds to the ions or molecule from one side of the membrane and transport to the other side. What are the carrier proteins? Carrier proteins are the transporter proteins or transporters which binds ion or molecule from one side. एका साइडला मेम्ब्रेनच्या either outside or either inside. It binds to the ions or molecules from the one side of the membrane and transport it to the other side, that is either from outside to inside or from inside to outside of the cell. That is, these are the carrier proteins or transporters which allow the passage of ions. So there are the three types of the carrier proteins: uniporters, symporters and antiporters. Uniporters, symporters and antiporters. So what is uniport? The transport of only single molecule across the membrane is the uniport. Mechanism by which there is a transport of only single molecule across the membrane. Transport of only single molecule across the membrane is known as uniporter or uniport mechanism. So glucose and amino acid transport in the cell, erythrocytes glucose transporter transport glucose in the cell. This transport does not require, that is, this uniporter does not require any kind of ATP energy for the transport of molecule. See, only one molecule is transport inside is known as the uniport mechanism. That is, the single molecule crosses the cell membrane. This is the cell membrane, and this is the protein which is present in the cell membrane, and this protein transport only single molecule from outside to inside the cell. Now, what is Symport? Symport is the type of co-transport system. आताच आपण सोडियम ग्लुकोज सिम्पोर्ट शिकलो. It is the type of co-transport system, that is the transport of two substrates in the same direction, is called as Symport. See here, this is one molecule and this is second molecule. These two molecules are transferred inside the cell through this cell membrane. This protein is located in the cell membrane. This protein allows the passage of two molecules inside the cell by the Symport or co-transport mechanism. So transport of two substrates in the same direction is called as Symport. It uses energy generated during the primary active transport. Symportला काय असतं की एनर्जी लागते. That energy is generated during the primary active transport or electrochemical gradient from protons to transport molecule. It uses energy generated during the primary active transport or electrochemical gradient generated or electrochemical gradient from protons which is generated during the transport of molecule. Example, amino acids, glucose transported by Symport mechanism. Symport mechanism आत्ताच just we have seen the mechanism, that is sodium glucose Symport, that is simultaneous passage of sodium and glucose inside the cell. For example, amino acid, glucose, these are transported by Symporter mechanism. Okay, are you getting me? झोपली आहे का? 3 वाजले तर का लक्ष आहे तुमचं? अरे काय प्रेझेंट 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 प्रेझेंट. ओके. फिजिकली काही प्रेझेंट राहू नका, लक्ष द्या इथे, मेंटली पण. काय शिकवलं आता? Symport. That is the transport of two molecules. Symport is also known as co-transport. That is simultaneous passage of two molecules inside the cell. Are you hearing me properly? Okay, Ashwini, I have answered very good. Okay, so I'm going to continue it. So it is the type of co-transport mechanism. That is the transport of two substrates in the same direction. That is the symport. And next is antipodes. So when the two substrates are moved in the opposite direction across the lipid bilayer, this is the lipid bilayer, the process is known as the antipodes. That is, two substrates are moved in the opposite direction. One is from outside to inside, and another is from inside to outside. So when the two substrates move in the opposite direction across the lipid bilayer, the process is known as antipode. Two substances move in the opposite direction in antipode. Symport मध्ये two substances moves in the same direction, and uniport मध्ये only one substance moves in the only one direction. So antiporters couple the movement of one type of molecule against its concentration gradient with the movement of other ion down its concentration gradient in the opposite direction. म्हणजे एक मॉलिक्यूल जो आहे तो हायर कॉन्सन्ट्रेशन टू लोअर कॉन्सन्ट्रेशन, आणि दुसरा मॉलिक्यूल जो असेल तो लोअर कॉन्सन्ट्रेशन टू हायर कॉन्सन्ट्रेशन. मग हायर कॉन्सन्ट्रेशन टू लोअर कॉन्सन्ट्रेशन होताना काय होईल? एनर्जी इज रिक्वायर्ड, सो फ्री एनर्जी ऑफ एटीपी इज यूज इन द ट्रान्सपोर्ट ऑफ मॉलिक्यूल. याला हायर टू लोअर कॉन्सन्ट्रेशन कडे जाताना काय होईल? एनर्जी लागेल. मग एटीपी इज यूज ॲज अ एनर्जी सोर्स इन अ अँटीपोर्ट मेकॅनिझम. अँटीपोर्ट मध्ये एटीपी एनर्जी लागतं, तसं सिम्पोर्ट मध्ये पण एटीपी एनर्जी लागते. युनिपोर्ट मध्ये कोणत्याच टाईपची एनर्जी लागत नाही. युनिपोर्ट मध्ये म्हणजे फक्त पासेज फ्रॉम in only one direction, movement of only one molecule in only one direction. But symport is a movement of two molecules in the same direction, and antipode is movement of two molecules again in the opposite direction. So antipode is again the opposite direction. This is known as antipode. In very simple language, I am trying to convey you this topic. So antiporter is the mechanism in which one molecule moves against the concentration gradient while the another molecule moves along the concentration gradient. म्हणजे against the concentration gradient means from lower concentration to higher concentration. Okay, so this is the antipode is again one type of transport system in bacteria which require ATP as the energy source. Are you getting? समजला का काय काय शिकवलं? सिम्पोर्ट, सोडियम ग्लुकोज सिम्पोर्ट, त्याच्यानंतर टाइप्स ऑफ आयर्न ट्रान्सपोर्ट, त्याच्यानंतर अजून काय शिकवलं? Main first sodium glucose symport, that is the transport of sodium and glucose inside the cell by the sodium glucose symport mechanism. So this is the sodium glucose symport pump. त्याच्यानंतर कॅरियर प्रोटीन्स आर ऑफ 3 टाइप्स, युनिपोर्टर्स, सिम्पोर्टर्स आणि अँटीपोर्टर्स. Uniporters, that is in only one direction, transfer of molecule, transport of molecule. Symport, in the same direction, transport of two molecules in the same direction using the energy generated during the primary active transport is used as a energy in the Symport mechanism. And antiports uses the ATP energy as the energy source, that is transport against the concentration gradient and in the opposite direction, that is one molecule moves inside and another molecule moves outside of the cell. So our next topic is the group translocation. Group translocation. So what is group translocation? It is the type of active transport system in prokaryotes. Group translocation is type of active transport system. It transport molecule across the cell membrane with chemical modification. काय होतं की ग्रुप ऑफ मॉलिक्यूल और मॉलिक्यूल पासेस अक्रॉस द सेल मेम्ब्रेन विथ द केमिकल मॉडिफिकेशन. कुठल्या त्याच्यामध्ये मी कंपाऊंडमध्ये केमिकल मॉडिफिकेशन होतं असं सांगितलं नाही. ग्रुप ट्रान्सलोकेशनमध्ये काय होतं, ट्रान्सपोर्ट अक्रॉस द सेल मेम्ब्रेन विथ द केमिकल मॉडिफिकेशन. सो धिस ग्रुप ट्रान्सलोकेशन वॉज इंट्रोड्यूस बाय सायंटिस्ट मिशेल अँड मॉयले इन 1958. कोणी शोधून काढलं? मिशेल and Moyle in 1958. So the best known group translocation system is phosphoenol pyruvate sugar phosphotransferase system. कोणती बेस्ट ग्रुप ट्रान्सलोकेशन सिस्टम आहे? फॉस्फोइनॉल पायरुवेट शुगर phosphotransferase system in organism. It is the unique uptake of sugar in bacteria. The phosphoryl group donor is in this group translocation mechanism is not ATP but phosphoenol pyruvate is acts as a phosphoryl group donor. Phosphoryl group donor म्हणजे फॉस्फेट ग्रुप कोणाकडनं येतो? जनरली रिएक्शन मध्ये ATP कडनं. What is ATP? ATP is the adenosine triphosphate. So this adenosine triphosphate donates phosphate group generally in all metabolic reactions. But in this group translocation by phosphoenol pyruvate system, in phosphoenol pyruvate acts as a phosphoryl group donor for the transport of sugar. शुगर ट्रान्सफर सिस्टीम आहे ही कोणती? फॉस्फोइनॉल पायरुवेट शुगर फॉस्फोट्रान्सफरेस. म्हणजे या सिस्टीमचं नाव काय आहे? शुगर फॉस्फोट्रान्सफरेस सिस्टीम, PTS. Phosphoenol pyruvate sugar phosphotransferase system. So what are the components of PTS phosphotransferase system? It is the multi-component system, consists of 4 protein component. 4 protein component कोणते? Enzyme first, HPR, enzyme second A, which is present in the cytoplasm, and enzyme second B present in membrane. In some organism actually there are only 4. एन्झाईम फर्स्ट पहिलं आहे, HPR सेकंड आहे, एन्झाईम सेकंड ए तिसरं आहे, एन्झाईम सेकंड बी चौथं आहे आणि काहींमध्ये एक एन्झाईम सेकंड सी एक ॲडिशनल कंपोनंट प्रेझेंट असतो. So it consists of enzyme second C also, but mainly 4 component: enzyme first, HPR, enzyme second A, which are present in the cytoplasm, and enzyme second B, second C present in the membrane. काही ऑर्गॅनिझम मध्ये सेकंड सी असतो, काहींमध्ये नसतो. मग चौथे कोणता आहे? एन्झाईम सेकंड बी, विच इज प्रेझेंट इन द सेल मेम्ब्रेन. So what is mechanism of group translocation? Mechanism of group translocation involve transfer of the phosphoryl group from phosphoenol pyruvate to sugar. This takes place in sequential manner. With sequential manner, the phosphoryl group first transport from phosphoenol pyruvate to enzyme first. Enzyme then transfer phosphoryl group to heat resistant protein HPR to form phospho-HPR. The phosphoryl group is then transferred to sugar-specific enzyme second A. Enzyme second A, in the final step, the phosphoryl group is then transferred to sugar molecule. This reaction is catalyzed by enzyme second B, which is the sugar-specific. The sugar is released inside the membrane in the form of modified form as a sugar phosphate. आता मी तुम्हाला परत एकदा स्टेप्स सगळ्या समजून सांगते. आर यू गेटिंग मी? समजतंय का? Mechanism of group translocation. Now you pay your attention here. What is the mechanism of group translocation? Transfer of phosphoryl group from phosphoenol pyruvate to sugar. आता ग्रुप ट्रान्सलोकेशन हे कशासाठी आहे तर ट्रान्सपोर्ट ऑफ शुगर फ्रॉम आउटसाइड टू इनसाइड. बघा, शुगर म्हणजे ही ग्लुकोज. सो ट्रान्सपोर्ट ऑफ ग्लुकोज फ्रॉम आउटसाइड, दिस इज द सेल मेम्ब्रेन. Okay, this is the cell membrane, and transport of this glucose sugar from outside to inside of the cell, या मेम्ब्रेनच्या मध्ये पास करायचं आहे. मग ते हे कसं पास होईल? बाय द ग्रुप ट्रान्सलोकेशन. ग्रुप ट्रान्सलोकेशन म्हणजे काय? शुगर, ग्लुकोज शुगर अशी ज्याच्या मध्ये ग्लुकोजच्या फॉर्म मध्ये न जाता ग्लुकोज ट्रान्सफर होते इन द फॉर्म ऑफ ग्लुकोज 6 फॉस्फेट. Glucose transfer in the cell in the form of glucose 6 phosphate. So how this glucose 6 phosphate is formed? First, phosphoenol pyruvate donates phosphate group. This is the phosphate group to enzyme first. Phosphoenol pyruvate donates this phosphate group to enzyme first. Enzyme first is form the phosphorylated enzyme first. Phosphorylated enzyme first then donates phosphate group to HPR. What is HPR? HPR is the heat resistant protein. HPR म्हणजे heat resistant protein. So this enzyme first donate phosphate group to HPR. HPR then donates phosphate group to enzyme second A. हे enzyme second A आहे. It donates phosphate group to enzyme second A. Enzyme second A become phosphorylated. बघा काय? ट्रान्सफर ऑफ फॉस्फोरिल ग्रुप फ्रॉम phosphoenol pyruvate to sugar. It takes place in sequential manner. Phosphoryl group first transfer from phosphoenol pyruvate to enzyme first. Enzyme first then transfer phosphoryl group to heat resistant protein HPR to form phospho-HPR. The phosphoryl group is then transferred to sugar specific enzyme second A. This enzyme second A is the sugar specific enzyme. All these enzyme first, enzyme second A, HPR, these are present in cell cytoplasm. Enzyme second B is present in cell membrane. This is the cell membrane. Okay. this is cell membrane. So and glucose is present outside the cell, this is outside. So this enzyme second A donate this phosphate group to enzyme second B, which is present in the, or which is attached to the enzyme second C in the cell membrane. second B आणि second C हे सेल मेम्ब्रेन मध्ये असतात. हे फॉस्फेट ग्रुप हे एन्झाईम सेकंड बी ला अटॅच होतो. मग ग्लुकोज थ्रू दिस protein, through this enzyme second protein, it passes inside the cell in the form of glucose 6 phosphate आणि ग्लुकोज 6 फॉस्फेटच्या फॉर्म मध्ये हे ग्लुकोज मॉलिक्यूल सेल मध्ये पास होतं. So in the final step, phosphoryl group is then transferred to sugar molecule. The reaction is catalyzed by enzyme second B, which is the sugar specific. The sugar is released inside the membrane in the modified form as a sugar phosphate. Sugar कोणत्या फॉर्म मध्ये सेल मध्ये जाते? In the form of sugar phosphate. Okay, do you get me? समजलं का ग्रुप ट्रान्सलोकेशन? Have you understood the mechanism of group translocation, Jayashri? It is actually simple, very simple, and if you will learn the diagram, you will be able to write the description. There is no problem. So I will again show you the mechanism of group translocation. What is group translocation? It is the type of active transport system in prokaryotes which was introduced by scientists Michel and Moyle in 1958. It transport sugar molecule across the cell membrane with the chemical modification. तसं ग्लुकोज बाहेरून सेलच्या बाहेरून सेलमध्ये जात नाही. ग्लुकोजचं मॉडिफिकेशन होतंय. काय केमिकल मॉडिफिकेशन होतंय? ग्लुकोज इज ट्रान्सफर ॲज अ ग्लुकोज फॉस्फेट, म्हणजे शुगर इज ट्रान्सफर ॲज अ शुगर फॉस्फेट इनसाइड द सेल. And the best known example of group translocation is the phosphotransferase system. That is phosphoenol pyruvate sugar phosphotransferase system in E. coli. Sugar phosphotransferase system in E. coli. This is the best known example of group translocation. It is unique for the uptake of sugar in number of bacteria. So what is the role of this phosphotransferase system? ATP is not used as a phosphoryl group donor. Instead phosphoenol pyruvate is used as a phosphate group donor. Phosphate group कौन donate करते हैं? Phosphoenol pyruvate. Okay, what are the components of PTS? Enzyme first HPR and enzyme second A, which are present in cytoplasm. Enzyme second B, enzyme second C present in cell membrane. हे एवढ्या सगळ्या सिक्वेन्शियल स्टेप तुम्ही सगळ्या लिहू शकाल जर तुम्ही हे व्यवस्थित समजून घ्याल तर. व्हॉट इज मेकॅनिझम? फॉस्फोइनॉल पायरुवेट डोनेट फॉस्फेट ग्रुप टू एन्झाईम फर्स्ट. पायरुवेट इथून रिलीज होतं, एन्झाईम फर्स्ट फॉस्फेट ग्रुप होतं, एन्झाईम फर्स्ट बिकम फॉस्फोरिलेटेड. Enzyme first phosphorylated donate phosphate group to HPR, that is the heat resistant protein, which become phosphorylated phospho HPR. Phospho HPR donates phosphate group to enzyme second A, which is converted to phosphorylated enzyme second A. Enzyme second A donates phosphate group to enzyme second B, which is present in the cytoplasm, which is present in the cell membrane. The cell membrane protein enzyme second B binds to the glucose, and when it binds to glucose, it donates this phosphate group to glucose, and glucose is transferred in the cell as a glucose 6 phosphate, is known as the phosphotransferase system in E. coli. So sugar is released inside the cell in the form of modified and that modified form is the sugar phosphate. Directly शुगर सेलमध्ये न जाता कशी गेली सेलमध्ये, शुगर फॉस्फेटच्या फॉर्ममध्ये गेली. ओके. डू यू हॅव एनी डिफिकल्टी? आजचं जे जे शिकलो त्याच्यामध्ये काही डिफिकल्टी आहे का? पटापट चॅट बॉक्स मध्ये मेन्शन करा, अदरवाइज आय एम नॉट गोइंग टू स्टॉप माय स्क्रीन शेअरिंग. डू यू हॅव एनी डिफिकल्टी? ओके.